Hello. So we'll start our lecture today about the transport of ions and small molecules across cell membranes. And we will see what kind of molecule can transport via the cell membrane or the plasma membrane of the cell, and what kind of molecule that cannot transport. So movement of virtually all molecules and ions across cell membrane is mediated by selective membrane transport proteins. And the selective permeability, this one of the major characteristic of the plasma membrane. And this membrane transport protein, as we knew last lecture, it is embedded in the phospholipid bilayer. So few molecules can cross the membrane by passive diffusion, like gases, oxygen and carbon dioxide, small uncharged polar molecules like urea and ethanol. Such molecule also can diffuse across cell membrane without the help of any transport protein. Okay, and it's very obvious that the gases can transport without any need of help. It's passive diffusion because for the process of respiration as well urea for the process of excretion. So no metabolic energy is used or expended because of the movement is from the high concentration to the low concentration, which normally we say down its concentration gradient. So when we say down its a chemical concentration gradient, that's meaning molecules are moving from high concentration into low concentration. So this figure shows actually that gases, if you can see, gases can penetrate, can be transported easily. So the membrane is permeable to gases, while small uncharged polar molecules, you can see that ethanol is permeable, while water is slightly permeable, as well as urea, but it still can pass, while large uncharged polar molecules like glucose and fructose, they are purely impermeable to pass through the membrane. They need the carrier protein to transport these molecules, while ions as well is impermeable because it will change the ionic strength and the potential difference between the inside of the cell and the outside of the cell. While the charged polar molecules like amino acids, the ATP, glucose, 6-phosphate protein, the nucleic acid, of course, it's very permeable. Um, otherwise, it will leak out of the cell because these charged polar molecules is found majorly inside the cell as well. So it must be impermeable. So the diffusion rate of any substance across a pure phospholipid bilayer is proportional to its concentration gradient across the layer and the hydrophobicity as well as the size and the charge. So four parameters control if the molecule will pass or not pass through the plasma membrane. So the membrane permeability can be easily determined by adding some radioactive material. So if you want to investigate if a molecule can pass through a plasma membrane or cannot, you can label this molecule by radioactive material, and you can track its movement from one compartment, and you can measure the rate of appearance in the other compartment. That means moving either from the outside of the cell to the inside of the cell to the cytoplasm, or even measure the rate of diffusion or the rate of movement between the cytoplasm and the nucleus to the inside the nucleus through the nuclear envelope. So the greater the concentration gradient of the substance, of course, the faster its rate of diffusion across a bilipid layer. So the hydrophobicity, that's mean the hydrophobicity, that's mean how the molecule is not soluble in water. So the hydrophobicity of a substance is measured by a coefficient which is called partition coefficient K. So the higher K, the more lipid soluble it is. So if the molecule have a higher K, that means it's a very lipophilic. This is the reason that the more hydrophobic a molecule is, the faster it diffuses across a pure phospholipid bilayer. So a hydrophobic molecule is the same as a lipophilic molecule, okay? So the higher hydrophobic molecule, the higher lipophilic molecule, the higher it can pass across a pure phospholipid bilayer. And this molecule will be characterized by high partition coefficient K. For example, diethylurea has a K of 0.01, whereas urea has a K of 0.0002. So in this case, diethylurea will be is actually 50 times more hydrophobic than urea. So obviously it will diffuse through the phospholipid bilayer about 50 times faster than urea. So if a transported substance carry a charge, now we are talking about molecules that have a charge, its movement also is influenced by its concentration gradient and the membrane potential. That's mean the balance of the charge outside and inside the membrane. So the electrical potential voltage across the membrane and the combination of these two factors, the electrochemical, the concentration gradient and the charge over the molecule, we term it electrochemical gradient. So let's talk now about membrane proteins that mediate the transport of most molecules and ions as well across biomembranes. So the transport of most molecules into and out of the cells require the assistance of a specialized membrane protein, which is actually the integral protein we have talked in the last lecture. So molecules with relatively large K, example as water and urea, is accelerated by a specific protein because their transport by passive diffusion usually is not sufficient and is not rapid enough to meet the cellular needs. In other words, although water and urea can pass through the membrane by passive diffusion, sometimes they need a specific protein to accelerate this transport, as we said, because maybe the passive diffusion will not be enough to meet with the cellular needs. So the transport proteins allow movement of hydrophilic substance without their coming into contact with the hydrophobic interior of the membrane. So in other words, it's like a channel will be formed of the protein so that the molecule will pass through this channel and will never meet the hydrophobic core of the phospholipid bilayer. For example, ATP-powered pumps are ATPases that uses energy through the ATP hydrolysis to move ions or small molecules across the membrane, but against a chemical concentration gradient, or against an electrical potential, or against both of them. This process is called active transport. So now we understand now that active transport majorly differs than passive transport in the way that it uses energy from the breakage of the ATP. And usually the cell uses active transport in cases that it wants to move the ions or the molecules against its concentration gradient. That's mean it moves from the low concentration into high concentration. So channel proteins transport water or specific types of ions and hydrophilic small molecules down their concentration gradients or electrical potential gradient. Such a protein assists transport. Sometimes it's referred as facilitated diffusion. So now we have another type of diffusion, which is facilitated diffusion. So basically, facilitated diffusion, it is the same as passive diffusion, but in facilitated diffusion, there is a protein involved in the membrane that facilitates the transport, but it is still with the concentration gradient. It is not against the concentration gradient, and it is still, there is no energy use for this process. Some sort of channels, protein channels, is called non-gated channels, and these channels are used for some ions to be transported, and they are open much of the time. While gated channels is used by most other ions, and it opens only in response to specific chemical or electrical signal that lead to the opening of these channels. That's why it's called gated channels, and permitting the ion to either enter or exit from the cell. So the transporters The protein transporters, which is actually as well integral proteins, are also called carriers. They move a wide variety of ions and molecules across cell membranes. There are three types of transporters have been identified up till now: a unitransporter, an anti-transport, and a unit transporter, uniporters, and antiporters and symporters. Of course, the major difference, as the name implies, uniporters only transport one molecule, like glucose or each amino acid, while the antiporter, it moves two molecules in the opposite directions, while symporters, which is co-transport, it uses the energy driven from one molecule to move the other molecule, as we will see now. So ATP pumps use energy from hydrolysis of ATP, whereas co-transporters uses the energy stored in electrochemical gradient. And that's why it's termed secondary active transport. It's not purely active transport, okay? Because there is no energy is derived from the hydrolysis of ATP. So in these kind of carrier proteins or the transporter, in order to do its function, they must do conformational changes. And what is conformational changes? Actually, it is a change in the shape of protein to permit the movement of the ions across the membrane. So these conformational changes are essential to the function of all transport protein. The ATP-powered pumps and the transporters undergo a cycle of conformational change, exposing a binding site or sites to one side of the membrane. This site at one side of the membrane will accept the ion or the molecule, and then this is followed with another conformational change to permit the release of the ion or the molecule on the other side of the membrane. Okay, as we will see in the figure now. Later, ion channels as well shuttle between a closed, because it's a channel, shuttle between a closed state and the open state. But many ions can pass through an open channel without any further conformational change. It's obvious because it's a channel, so it doesn't need it, just open and close. Let's see how this works. So here we can see the difference between different type of carrier proteins. In the first, let's have a pointer here. So here you can see that this is ATP powered pumps. Why it's ATP powered pump? You can see because there is a breakage of hydrolysis of ATP into ATP, and this hydrolysis will give the enough energy for this red molecule to pass through the carrier protein against its electrochemical gradient, electro ECG, electrochemical gradient. So it's against its concentration, and you can see from the arrow, it indicates that the higher concentration is outside of this molecule and the low concentration is inside. So in this case, the ATP powered pumps can transport up to 1,000 ion per second against its electrochemical gradient. While normally the ion channels at this type as well it's a kind of facilitated diffusion. So you can see here the ions moves with concentration gradient, from high concentration to low concentration, and you can see the ion channel can be closed, and then it can be opened for the release of the ions. And these ion channels, it can transport up till 100 million ions per second. While for the transporter, if you remember, we have three types: the uniporter, the symporter, and the antiporter. You can see that the uniporter only transport one ion. These uniporter proteins or transport carrier proteins only transport one ion through the concentration gradient. So it moves with concentration gradient. While the symporter, you can see as well, in the symporter, you can see this carrier protein, sim, that's mean with. Sim, it's a Latin word, means with. So you can see here that these two ions, one of them moves with the concentration gradient, you can see, with the concentration gradient, while the other surprisingly moves against its concentration gradient. If you can see, It moves from the low concentration into high concentration. So how this molecule is moved from the low concentration to the high concentration, and there is no ATP hydrolysis. Actually, as we said, the movement, the movement of this molecule from the high concentration to low concentration will drive some energy which will be enough for uh, recruiting and helping in the transport of this molecule from low concentration into high concentration. While the last type is the antiporter, and you can see it's the same principle. Actually, the main difference that the two molecules are moved into opposite direction. The red molecule is moved in this direction from outside to the inside, while the black molecule is moved to the outside. And the red molecule is moving with concentration gradient, and the black molecule is moving against the concentration gradient. And as well here, energy is obtained from them, as we said here, whereas co-transporters, and these cases are co-transporters, use the energy stored in the electrochemical gradient, and both of these are termed secondary active transport. So this table actually summarizes the mechanism of transporting ion and small molecules across cell membrane. So you can see it describes which of the what is the transport mechanism, and if it's requires specific protein, if the solute or the molecule is transported against its concentration gradient, if it's coupled to hydrolysis, ATP hydrolysis, and you can see only active transport that is coupled to ATP hydrolysis. If the driven by movement of a co-transport on ion down its gradient, okay, and as well examples for each type and which type of molecules. So you can see glucose and amino acids for symporters, various ions and sucrose for antiporter, ions, small hydrophilic molecules, lipids for the ATP powered pumps, glucose and amino acids for uniporters facilitate diffusion, while some ions and water as well for channels. Of course, gases, steroid hormones, and a lot of drugs are passively diffusion. You can see that co-transport sometimes is called a secondary active transport. So let's see some feature that distinguish uniport transport from passive diffusion. So the first feature is the rate of facilitated diffusion by uniporter is much higher than passive diffusion through a pure phospholipid bilayer. Because the transported molecule never entered the hydrophobic core of the phospholipid bilayer, so now the partition coefficient is irrelevant, the K, the partition, because actually partition coefficient K actually is calculated if is relevant if the molecule passes by its own self through the phospholipid bilayer. But since the molecule is passing through a transport protein, so it is irrelevant to have the value of the partition coefficient K is irrelevant. The third feature is that the transport occurs via a limited number of uniporter molecule rather than throughout the phospholipid bilayer. In other words, if the molecule passes through the phospholipid bilayer, it can pass from any site all over the cell. But if it passes by a uniporter, the number of these uniporters on the cell membrane are limited. So because so it can pass only through the uniporter molecules. So for this case, there is a maximum transport rate, Vmax, that is achieved when the concentration gradient across the membrane is very large and each uniporter is working at its maximum rate. So if you have 10 uniporter protein on the surface of the cell, so once the 10 uniporter protein are working and the transporting protein, and this will roughly this will roughly reach the Vmax. No more molecule can be transported since all the 10 uniporters are occupied. So Another feature important for uniporter that the transport is specific. So each uniporter transport only a single species of molecule or a single group of closely related molecules. So the measure of the affinity of a transporter for its substrate is termed Km, which is the concentration of the substrate at which the transport is half maximum. These properties also apply to transport molecules by the other classes of protein. Okay. So let's talk about first example, which is the glucose transporter GLUT1. GLUT1. It is the best understood uniporter. Actually, it stands for glucose unit transporter 1. So GLUT1 found actually in the plasma membrane of red blood cells. And of course, as you know, that RBCs are bags actually of hemoglobin. There is no nucleus, and it's a bag of that contain hemoglobin and surrounded by a plasma membrane. So you can see from the figure, here is the GLUT1. It exposes a binding site for glucose to the outside, and you can see that the glucose have high concentration to the outside more than to the inside. So this actually, it is the outward facing conformation shape. So it accepts the glucose, it is transformed to the inward facing conformation to release the glucose to the inside of the cell. And then it goes back to the outward facing conformation to repeat the cycle. So this actually is a very good example for the uniporter transporter in which is used for moving the glucose along with the concentration gradient. That's mean with the concentration gradient. So we can isolate and purify any transport this GLUT1 from mature erythrocyte is a straightforward procedures. GLUT1 actually uniporter transport glucose in most mammalian cells. And most mammal cells, of course, use the blood glucose as the major source of cellular energy. Like other uniporters, GLUT1 alternate, as we have just said, alternates between two conformational states. In one, which is the out-facing conformation, it is which have the glucose binding site faces to outside of the membrane, while the inward-facing conformation, in which the glucose binding site is facing the inside. In addition to glucose, which is transported by GLUT1, D-mannose and D-galactose are also transported by GLUT1. GLUT1 actually accounts for 2% of the protein in the plasma membrane of erythrocytes. So 2% of the total protein of the total integral proteins that present in the plasma membrane of erythrocytes are actually GLUT1. After glucose is transported into the erythrocyte, it is rapidly phosphorylated to form another molecule, which is glucose 6-phosphate. And this for two reasons. The human genome encodes a family actually of sugar transporting GLUT protein. So the GLUT actually, it's not only GLUT1, you have GLUT up till now, the discovered or identified isoforms of GLUT are GLUT1 to GLUT12. So we have 12 members in the family of glucose uniporter transporters. They are highly homologous in structure, and all are thought to contain 12 membrane-spanning alpha helix. So GLUT2 actually expressed in liver and the insulin secreting beta cell of pancreas, while GLUT4 is expressed only in fat and muscle cells. And these cells respond actually to insulin by increasing the uptake of glucose, thereby they help in removing glucose from the blood. As you know, the glucose can be stored in a fat cell or a muscle cell majorly, or liver as well. In the absence of insulin, GLUT4 actually is found intracellular membrane, not on the plasma membrane. So once the glucose insulin causes these GLUT4 vesicles, rich vesicle, this GLUT4 to be recruited to the plasma membrane, to fuse with the plasma membrane, and the increasing number of GLUT4 molecule on the cell surface will permit the larger higher rate of glucose uptake. So the effect, any defect in this process will lead, of course, to diabetes. For GLUT5, it transports actually fructose. You can see as well there are other GLUT family that have been other GLUT members in the family, and they are not have been studied in details because we just only talked about GLUT1, GLUT2, GLUT4, GLUT5. So the other members are not fully understood. So let's study how scientists can study the functional properties of transport protein experimentally. So the functional properties of these protein, which are the transport protein, can be studied only if they are associated with the membrane, because it's no logic to study a transport protein by its own self. It needs to be associated with the membrane to understand its function and to know which molecule it transports. Most cellular membranes contain many different types of transport protein. Making functional studies of a single protein is very difficult. So actually you can't study a transport protein. It's very difficult because normally the cell have a lot of transport protein on its surface. So to facilitate such a study, researchers usually use either of two approaches for enriching a transport protein of interest so that it predominates in the membrane. In one common approach, a specific transport protein is extracted. We can extract this protein, purify this protein, and this purified protein is then reincorporated into a pure phospholipid bilayer, such as liposomes. So you can see here the figure. Normally, GLUT1 is existing in the membrane together with other transport proteins. Then we can disrupt this membrane, extract this transport protein by using of a detergent and some buffer solutions, and then we mix it with a phospholipid micelles. So you can see the phospholipid micelles, and these phospholipid micelles together with the transport protein will arrange itself, as you can see here, and the transport protein will be incorporated in the membrane. And now we can study which type of molecule can be transported by this transporter. Of course, we are giving here example of GLUT1, but we are talking about if researchers want to know the function of unknown transport protein. In other approach, the gene encoding a specific transport protein can be expressed at a high level in a cell type that normally don't express it. So we take so for example, let's see, for example, if we don't know the GLUT4, if we discover that there is a transporter protein in fat and the muscle cell, for example, GLUT4, but we already don't know it is a GLUT4. So we isolate the gene of that GLUT4 from the fat and muscle cells, and then we induce the expression of this GLUT4 in a liver cell, okay? And then we compare between the normal function of a liver cell and the liver cell that have been that is expressing this GLUT4 transporter protein. And if we can see that there is a peculiar molecule have been transported in these overexpressed GLUT4 liver cells, that means this is the function of the GLUT4 of this unknown transporter in this liver cell. So why we express the GLUT4, for example, in this liver cell? Because normally we have said that GLUT4 is only expressed in fat and muscle cells. So we understand already that it is not expressed in liver or beta cell of pancreas. So for co-transporter, as you can see the figure as well, this is three figures representing the different transport mechanism. Uniporter transport only one molecule, symporter transport two molecules in the same direction, antiporter transport two molecules in opposite direction. And you can see that these one are called the coupled transport or secondary active transport. So for co-transporter, bisimporter, or antiporter, these are a class of proteins that transport ions and small molecules such as glucose for uniporters and glucose and amino acid we're talking now about the co-transport, against its concentration gradient. So co-transporter use the energy, as we said, stored in the electrochemical gradient of sodium or hydrogen or proton ions to power the uphill movement of another substrate. So we understand now that the two molecules which are transported in a co-transporter, either a symporter or an antiporter, one molecule of these two molecules have to be an ion, because the movement of this ion will generate the power uphill. Uphill, that's mean movement of a lower concentration into a higher concentration. An important feature of such co-transport is that neither molecule can move alone, so they have to move together. So co-transporters share some features with unit transporters, such as GLUT proteins. The two types of transporter exhibit certain structure similarity, as you can see from the figure, even they are close in structure. They undergo as well cyclic conformational changes to allow the transport during transport of their substrate. So let's see here an example of the transporter of one glucose with two sodium ions. And that's a symporter, so both of them will move in the same direction. So you can see here, initially, the sodium is outside higher concentration, inside lower, and the glucose is vice versa. So I have the outward facing conformation shape, which will accept the two sodium ions and the glucose. Once accepting these molecules, it will be transformed to inward-facing conformation, releasing the sodium and the glucose ion to the inside. And then it will cycle back to the outward-facing conformation. So it is clearly here you can see that glucose have been moved against its concentration gradient, while sodium have been moved to the with the concentration gradient. So the movement of the sodium, as we have just said, the movement of sodium will generate energy, which will drive the uphill movement of glucose against its concentration gradient. While for the another type of transporter, which is the antiporter, it transports three sodium ions together with one calcium ion. So you can see here that both of them are moved in opposite direction. That's why it's antiporter. And you can see that one of the ions is moved with concentration gradient, the red one, and the other moved against concentration gradient. To have more understanding, so you can see that for the three sodium, one calcium antiporter in cardiac muscle, so this antiporter plays a principal role in maintaining a low concentration of calcium in the cytosol. Keep in mind that the existence of calcium in the cytosol is very important to be regulated. Calcium is a very important ion that trigger a lot of signaling pathway inside the cytoplasm. So it has to be tightly regulated. So a rise in, as you can see, a rise in cytosolic calcium concentration in cardiac muscles triggers contraction. So if it's not, if the calcium in the cardiac muscle is not tightly regulated, the heart will do excess contraction. By lowering cytosolic calcium and the operation of the sodium calcium antiporter reduce the strength of heart muscle contraction. So you can understand now this antiporter is a very important protein for controlling the heart muscle contraction. Note that the movement of free sodium ions is required to power the export of one calcium ion against its concentration gradient to the extracellular medium. But keep in mind that calcium actually is not going outside the muscle cell. Actually, calcium is stored in the endoplasmic reticulum. Why it's stored in the endoplasmic reticulum? In case the cell uses more calcium. So let's talk now about the ATP power pump. So we talked about the passive diffusion, we talked about the uniporter, we talked about the symporter, we talked about the antiporter. Now we are talking about the fifth type, which is the ATP power pump. So ATP power pump actually transport ions and small molecules against their concentration gradient. So it's active transport. All ATP power bombs are transmembrane protein, integral protein with one or more binding site of ATP, and ATP actually bind to the cytosolic phase of the membrane, to the cytosolic part of the transmembrane protein. They normally do not hydrolyze ATP unless ions or other molecule needed to be transported. The energy from the hydrolysis of ATP, as we have just said, is used to move ions or other molecules uphill against electrochemical gradients. So actually there are four classes of ATP power pump. PFV classes transport ions only, while ABC classes class of ATPs is primary transport small molecules. So let's see here an example of P class. Again, we have PFV and the ABC class. So we have four classes. So for the P class ion, all P class ion actually consists of 5 subunits. So you can see from the figure, it's a 5 subunits, alpha, beta, and there are 3 subunits here. And you can see that the ATP binding site on the cytosolic phase of the transport protein. And you can see as well that alpha subunit have 3 binding sites for 3 molecules and 2 binding sites for 2 other molecules. So during the transport process, at least one of the subunit is phosphorylated. That's why it's called P class. Okay, so you can see the phosphorylation occur in the, on this subunit. So the transported ions are thought to move through the phosphorylated unit. So actually it moves here in this channel. So these class, the example of this class include the sodium-potassium ATPases on the endoplasmic membrane, which maintains low cytosolic sodium and high cytosolic potassium concentration, and this is typical actually of animal cell. Another example is the calcium ATPases, which actually pumps calcium ions out of the cytosol into the external media. While another example is calcium ATPs as well, but these calcium ATPs actually pump the calcium from the cytosol into the endoplasmic reticulum or into the smooth endoplasmic reticulum. While the proton potassium ATPase is actually, it's existing in the acid secreting cells of mammalian stomach, which produce the HCl. And as well, its main function to transport the protons, or actually the hydrogen ions, out to the stomach lumen, and as well potassium ion into the cells. Okay. So let's see another example of P-class ion bonds, which is a muscle. Actually, we'll talk about the muscle calcium ATPase. One of the important ATPases is calcium in the muscle, calcium ion channel in the muscle. So in skeletal muscle, calcium actually is crucial for the process of contraction and relaxation. So in skeletal muscles, calcium ion are concentrated and stored in a smooth endoplasmic reticulum. Actually, it's not only in skeletal muscles. Calcium always stored in the smooth endoplasmic reticulum. So the release of stored calcium ion from the smooth endoplasmic reticulum into the cytosol will triggers a contraction of the skeletal muscle. So calcium ATP is located on the SR membrane of a skeletal muscle, will pump calcium from the cytosol into the lumen, thereby will induce relaxation, which actually the vice versa process. So the current model for the mechanism of action of calcium ATPases in the SR membrane involves two conformation states, actually termed E1 and E2. Actually, it is a similar principle of what we have just said about the uniporter, about the co-transporter. If you remember, we have a conformational change, a conformational shape that have outward shape, and another one which have inward facing conformation shape. It's the same here, but here it's termed E1 and E2. Okay, let's see how it works. So you can see here, initially, let's take it, let's have a pointer. So you can see here is the calcium outside in the lumen of SR, smooth endoplasmic reticulum. It is the storage, so it have high concentration more than the cytosol. So you can see here this conformation, this actually the general structure of the calcium ATPases. These are the two calcium binding sites. This is the ATP binding site, and actually this is the phosphorylated subunit, and it is phosphorylated on the aspartate amino acid. So initially you have E1 shape, which is ready to accept calcium from the cytosol. And once it accepts the calcium from the cytosol, the calcium and the ATP hydrolysis, you can see here, the ATP binds here and the calcium is already bound to the sites. Then the phosphorylation of the ATP on the aspartate amino acid in this subunit will generate energy enough for the conformational change to switch to the E2 form from E1 to E2, now exposing the calcium ion to the lumen of the SR. So now it is storing, actually storing the calcium inside the smooth endoplasmic reticulum. This is followed by a process of dephosphorylation as well. So that's why removing of the phosphate, and the process of removing of the phosphorylate as well will generate some energy that is used by the E2 form to go back to the other conformational shape, which is the E1 form. So you can see as well, these are these sites actually is a very high affinity for calcium. And this as well, the site for the ATP binding. This will, as we said, will generate the E1P form that will, the second conformation shape, which is a E2, is characterized actually by low affinity binding site. That's why it will release the calcium ion. And as we said, the dephosphorylation will generate energy, enough for the returning to the E1 conformation shape. So let's talk about another example of the P class, which is the sodium-potassium ATPases. As you can see from the figure, is different, a little bit different. So now we are talking about two ions. The other one we're talking about calcium only. Here we're talking about sodium and potassium. And you can see that where sodium is located. So it's the sodium is higher in the outside, while the potassium is higher in the inside. And you can see the three binding sites of the sodium and the two binding sites of the potassium. And as well, have these pumps have the same general structure, which is 5 subunits. So this is a second important P class ion pump, and present in plasma membrane of all animal cells. And it's crucial for maintaining the sodium and potassium concentration in the intracellular concentration of sodium and potassium in animal cells. It moves the 3 sodium ions out and the 2 potassium ions into the cells. Of course, coupled by ATP hydrolysis. The mechanism of action is very similar to that of the calcium pump, except that ions are pumped in both directions across the membrane. In its E1 conformation, the sodium-potassium ATP have 3 high affinity sodium and the 2 low affinity potassium binding site. And of course, it will be vice versa in the E2 conformation. And you can see from the figure here, in the E1, it will accept the 3, have a high affinity for these 3 sodium ions. it will take the sodium ions, the phosphorylation process on the aspartate on this subunit will drive the energy of conformation to change from E1 to E2 at that site, at this conformation shape. The three sites now will have low affinity for the sodium, so it will release sodium and will have now high affinity for potassium, and the potassium will bind to the binding site. After that, the process of dephosphorylation will generate enough energy for the E2 complex to do a conformational change and go back to the E1 complex. So it will release the calcium to the interior, to the cytosol, and then it will go, it can accept more sodium and so on and so forth.
Yes, doctor.
So we talk now about the fourth class of ATP power bombs, actually, which is the ABC bombs. And ABC actually stands for ATP binding cassette. And it is a superfamily, includes several hundred different transport proteins found in organisms ranging from bacteria to human. And you can see that it consists of 4 subunits, 2 T's and 1 A, and 2 A's, and the A subunit is used for, it is the binding site actually of the ATP. Each APC protein is specific for a single substrate or a group of related substrates. So if it's iron, sugars, amino acids, and etc. So all ABC actually, all ABC transport protein consists of 4 core domains, 2 transmembrane domains, the T domain, which actually allow the passageway of transported molecules, and 2 cytosolic ATP binding A domain. Okay. Let's see here how the molecule now will be transported in the by this transporter. So we'll talk about the example, which is MDR1, and MDR1 is named according to, because it's a multi-drug resistant transport protein. So this multi-drug resistant, it's actually a family of proteins. It's a lot of different proteins, but all under the name of multi-drug resistant transport protein. So it have different isoforms. So MDR1, its discovery actually came from studies on tumor cells, and culture cells that exhibit resistance to drug. Such cells express elevated level in MDR1. In other words, scientists found that when tumors are treated with a certain kind of drugs, these MDR1 proteins have the ability to transport this drug outside from the cell to the exterior. That this will lead to a tumor cell that is resistant to the drug. So scientists understood that tumors that are resistant to therapy have to express the MDR protein on its surface. And these MDR actually found naturally in our liver cells, because liver cells actually, it is the site of detoxification in our body. So it have been equipped, liver cell, to have multi-drug resistance proteins on its membrane to release the toxic components outside of the cell. So this protein actually uses energy, of course, ATP, to export a large variety of proteins, a large variety of drugs from the cytosol to the extracellular medium. The MDR1 gene is amplified obviously in multi-drug resistant cells, resulting in a large overproduction of MDR1 protein. That's why tumor cells are sometimes resistant to therapy, resistant to drugs, because actually the drug is not allowed to stay inside the cell because it is expelled to the outside by the multi-drug resistant protein on the membrane. So most drugs that are transported by MDR1 are small hydrophobic molecules. Two such drugs are colchicine and venlafaxine, and actually these are very two important drugs used for the treatment of cancers because they block the assembly of microtubules. So if the cell have a multi-drug resistant protein on its surface, this drug will not have an efficient function to block the assembly of microtubules. And of course, if the drug blocks the assembly of microtubules, actually it will block, obviously it will block the formation of spindle fibers. And the spindle fiber actually is crucial in the process of division of the cancer cell. So export of such drug by MDR1 reduces their concentration in the cytosol. As a result, a much higher concentration of drug is needed to kill cells that express MDR1 than those that do not express. So there are 50 different mammalian ABC transporters are now recognized. These are expressed in abundance in liver, intestine, and kidney. Actually, these are actually the sites where natural toxic and waste products are removed from the body. So it's very obvious that they are highly expressed in these cells. Substrates include sugars, amino acid, cholesterol, peptide, protein, toxins, and xenobiotics are the molecules that can be worked on by MDR. The normal function of MDR1 is to transport nature and the metabolic toxins into bile, intestinal lumen, or forming urine. So tumors derived from MDR expressing cells such as hepatomas, liver tumor, frequently are resistant to virtually all chemotherapeutic agents because they express MDR. Thus, it's difficult to treat because the tumor exhibit increased expression of MDR1. So the substrate actually for MDR1 of mammalian MDR1 are primary must be planar, not a 3D. Must be lipid soluble molecules because you can see it's not a channel. You don't have a channel. So the mechanism of transport by MDR1 is a special mechanism. It's called the Flippase model. So in this Flippase model, according to this model, MDR1 uses energy, ATP, and flips the charged substrate molecule from the cytosolic to the exoplasmic phase. There are another molecule, the MDR2, has been shown to flip phospholipids from the cytosolic facing region of the plasma membrane to the exoplasmic region. Thereby, it will generate an excess of phospholipid in the exoplasmic region. These phospholipids then peel off into bile duct and form essential part of bile. So here we can understand how the molecules are transported in MDR1. So here the molecule is a lipid, you can see head and tail, and it is planar. So it's not like 3D structure, it should be planar. So it starts here, the charged end with it have a charged end and hydrophobic end. So you can see it start to insert, the molecule start to insert itself into the cytosolic phase of the membrane, and then it is picked up by the transporter. You can see it picked up between the two T domains, two transmembrane domains, and now we can see the molecule is flipped. So to have the head part to the outside. Once it is flipped, it is released to the phospholipid bilayer, so it can be expelled to the outside. And this concludes our lecture for today.